For this particular meeting, I spoke about our international breast cancer research, where it's actually an organization called the International Center for the Study of Breast Cancer Subtypes. I serve as the scientific director. We conduct breast cancer research across the African diaspora, specifically with the intention of identifying genetic and molecular phenotypes that are specific to the West African diaspora, and hopefully to explain some of the disparities in cancer mortality that we see in breast cancer. So today I talked about the organizational structure, some of our sort of infrastructure in collecting samples, the partnerships we have with our African countries, the capacity building we do there, and then I also presented some emerging findings from the work that we do with the cohort. So I will be remiss not to mention that I work with the Chief of Breast Surgery, Lisa Newman. We were both recruited there this past year. And she, over the last 17 years, has been working with various teaching hospitals throughout Ghana. She also works with a teaching hospital in Ethiopia. So the sites in Ghana include Kumasi Teaching Hospital and some of its satellite clinics. In Ethiopia is St. Paul's Millennium Institution or Medical College, and also some of their satellite hospitals and clinics as well. We have site coordinators and site principal investigators. Typically, the site principal investigators will be the chief medical officers and the chief of surgery for those institutions. And then we also work with the departments of pathology to provide reagents to upscale or augment the quality of their tissue preservation and subtyping for tumors as well. Right, so as a geneticist, one of the things that I'm very interested in is how the shared West African genetic ancestry of the African Americans in the United States, because the large majority of the African Americans in the United States are descendant of African slaves that were brought over from the transatlantic slave trade. And so I'm interested in sort of identifying some of the shared ancestry predispositions that come hand in hand with our West African ancestry. So Dr. Newman previously observed and published prolifically on different phenotypes that she saw from surgeries she performed in patients who had breast cancer throughout West Africa. And then when she compared those phenotypes to East Africa, she saw that there was a distinct pattern of tumor biology in women in West Africa currently that also paralleled the tumor biology that we see in African Americans in the United States. So for instance, triple negative breast cancer is the most prevalent subtype of breast cancer that's diagnosed in African American women in the United States, and I think if you look across the board, it's probably about 40% chance that if a black woman has breast cancer, it'll be triple negative. In West Africa and Ghana and Nigeria, for instance, it's upwards of 80%. So the predominant type of breast cancer that's diagnosed in West Africa is triple negative. And so what we started to uncover are what we call population-specific alleles in certain genes. So these are mutations in genes that exist only in that population in sub-Saharan West Africa that are linked to predisposition for triple-negative breast cancer. One of those genes is called DARC, D-A-R-C. And so the title of my talk today was The Dark Side of Breast Cancer, because I'm a sci-fi fanatic. And what we study about the DARC gene is the fact that there is a very famous mutation called Duffy null in the gene, and it confers a resistance to certain species of malaria pathogens. And so it's fixed in sub-Saharan Africa. The result of the mutation is that it's removed from red blood cells, which is why it's a blood group antigen. But what we identified in terms of cancer context is that tumors that express the gene have a better or just a higher prevalence of immune cell infiltration. And so we identified that on the RNA level using some transcriptomics. And so now what we're starting to do is look within the tumors on a spatial proteomic level to actually see that tumors that express dark have better infiltration in the tumor space, whereas tumors that don't express dark, though there may be tumor-associated leukocytes in the region of the tumor, in the microenvironment of the tumor, they are sort of sequestered or compartmentalized into the stromal area of the tumor, and they don't infiltrate into the epithelium. And so what we're trying to determine is if that is because of the Duffy null mutation, for instance, in the gene, does that track with whether or not the tumor is expressing the gene? And it appears it probably does, but we're still working on a mouse model, for instance, to definitively determine that. So I think that just generally as a scientist who is interested in disparities across an admixed population, it's very important to deconvolve the genetic diversity that's in the admixed population. So to date, most GWAS studies that look for genetic associations with breast cancer predominantly only looked at European ancestry populations. More recently, there have been some consortium that brought together smaller cohorts to create a larger cohort to replicate some of those GWAS studies of African Americans, for instance. And they, by far, have identified some alleles that appear to be associated with breast cancer in those populations. But mechanistically, they haven't necessarily been linked to a why, okay? And I think it's important that so some of the presumption behind that is it's linked to their West African ancestry, that the reason you could find these genetic associations in African Americans is because they have a shared ancestry, but that hasn't actually been connected directly to the ancestral population. So as a geneticist, I think it's very important that we don't presume, because we have the tools now to actually go to the ancestral population, identify the risks in that population, and if the theory is right that the shared ancestry in African Americans is what's causing triple negative breast cancer incidence to be higher in that population, then we should be able to identify it in the ancestral population and then see it imprinted in the admixed population. So I think it's important in that regard. But in terms of the clinical impact, immunotherapy is one of the most hottest, sort of therapies, pan-cancer, right? I mean, regardless of where your tumor is, everyone wants sort of this super immune therapy treatment that looks like, it's amazing when it works. But in some people, it doesn't work at all. And so while we sort of stratify the population to say, if it works for you, sorry, it doesn't work for you, I think it's important to identify the why, So beyond the fact that we know that certain tumors are more immunogenic than others. Is there a way to identify why tumors are more immunogenic than others? And if I can identify the genetic or a gene that's driving tumor infiltration, then that gives a platform for creating, or recreating that type of phenotype in a tumor. So we're a long way from doing that, but I think that eventually it will be feasible.